medical school. He graduated with a veterinary science degree from AP Agriculture University, Tirupati in 1986 and earned a master in microbiology and immunology from the same university. He received his PhD from Iowa State University, USA in 1994. He is on the Executive Council Board of the Federation of Asian Biotech Association. He is also a member of several other organizations that promote research in medicine and biotechnology. He has published 30 scientific papers in reputed journals including Nature, PNAS, Immunology, and PLOS. He also initiated the annual international conference on 3R advances and alternative, which attracts several reputed speakers and was invited by the World Congress in 2023. He is building the research ecosystem in India, aspire to create more entrepreneurs and startups, thereby creating innovative thinkers in biology and medicine. His startup, Akasta Health, work on creating a biomarker-based biological age and health span assessment platform, researching on aging degenerative condition for biologic therapeutics, and was presented on the 2024 Healthcare Company of the Year Award and Successpreneur Award, apart from being nominated as a top 10 startup by Pharma Outlook for 2023. He is actively involved in promoting biotechnology alternatives to animal in the scope of 3Rs and research in diagnostic and therapeutics. Held leadership position in LASA and continues to advise lab animal science program in India and across the world while actively promoting 3R principle and alternatives. He is a key opinion leader in the new approach methodology, programs, and 3D models of human disease. I now invite our speaker to begin the today's session. Thank you.
Thank you so much, Twinkle. I really appreciate whatever. I'm trying to show the screen share. Let me see.
It's visible. It is visible.
Visible, sir.
It's visible. I just want to share. Okay, can you see the screen now?
Yes, sir.
Yes, sir, it's visible.
Yes, sir, visible.
You
Okay.
need to put it in full screen mode.
I just did. Yeah.
Yeah, it's fine
Okay.
now.
Okay, thank you so much for inviting me to be the speaker today for enlightening all of us about my own experiences with 3D spheroids, organoids, and tumoroids. What I'm going to do today is how I happen to be a veterinary pathologist and a veterinarian, worked with a lot of lab animals, and then slowly switched over to the relevant model. I call this relevant because we use human cells, not animal cells anymore in the in vitro methods. Of course, I will talk about the challenges here, but of late what's happening is these are called new approach methodologies or alternatives to animal experimentation. FDA, European Union, and others have started accepting results coming from in vitro systems. I will tell you the reasons why they are doing it now and what happened. Okay. So basically, it's an in vitro cell culture system that we developed, and they look like spherical balls, like an ELISA plate here, and each one will have a spherical ball like this that you see in the red color that is stained for cell membrane. So let me go to the next slide. When I said we developed these models, we are talking about different diseases where we create a model in the dish. The dish is the well or the petri dish, but it can be 96 well, 365 well, 12 well, anything. So you can consider that as a dish, each well. And the spherical models that we have in each well are called the spheroids. So we do this to replace ineffective animal testing. I call animal testing ineffective because many drugs are tested on animals and 99% of them fail. 1% gets into becoming a drug. So I call it is not an effective way of discovering a drug. So we developed spheroid and organoid. I will tell the differences later. We use these models, 3D models, and also monolayers, which you are all probably familiar with cell culture. single cell culture layer that flatly grows on the glass surface. That's a monolayer. We also use that to test the drugs. So 3D alternative cell culture models are also called as non-animal methods. As a veterinarian, I used lab animals, but when I started OncoSeq Bio in 2019, I decided that I will use cell culture models for my uh, drug discovery, pesticide testing, new biologic testing, for safety, for toxicology, for efficacy, anything that you can tell. We can test it in the in vitro system. So let us see what are the models we have in the animals and what are we trying to replace here or reduce here. Because two things that happen in the animal model. Animal is not a human. We all know that. So there will be differences in the experimentation. When I said animal models, it could be a fly or an earthworm or a hamster, opossum, ferret, goat, monkey, mouse, hamster, pigeon, or a fish. And there are so many animals available for modeling, and each one has a specific anatomical feature or a characteristic that makes it a good model for a particular disease. For instance, if I pick a hamster, it's a good model because it has large cheek pouches and it stores the food there. And it also has an immune system around the cheek region that can be measured easily. So we use it for immunology research around the cheek region. And when I say we have monkey, then monkeys are heavily used for diabetes because it's very similar to human in the way it developed diabetes. For measles, it also gets affected by measles infection. So we have a metabolic infection for cardiovascular and also for vaccines. So that's the closest to the human. So we rely a lot of testing on monkeys. So like the dog is tested for safety, pharmacology, heart rhythms and all that stuff. And mouse and rat, of course, we test it for many toxicology and all that stuff. So, and earthworm and bees, we use them for environmental toxicity and other things. Zebrafish, we use for teratogenicity, embryonic, and of late, it has been used for many things in toxicology and water contaminants, and easy to administer, short lifespan, so on. So these are different models where we use them for a specific reason on their anatomical feature or their characteristic behavior or the response to a particular disease. These animals we want to replace or reduce because they are not humans. They don't have human cells. We just understand the physiology, but we cannot reciprocate or extrapolate it to humans. So the big statement I am going to make is human and animal biology may be similar, but it is not the same. So that is very important for us to recognize. So I have half a picture of a fly and a woman here, and then a cat and a rat and a dog, pig and a monkey. And they're all very similar. When you look at it, they're alive, they're able to move, they're able to behave, they're able to respond just like humans. But it is not the same, and we all know that. That's why probably so many drugs fail. So there is an opportunity for us to explore alternatives which could be better than animal models to assess safety. So when we tested many drugs in animals, here are a list of drugs on the right side. You can read them, but I will do it for you. Bextra, Becol, Resilin, Hispanol, Permax, Raxar, Paladon, Micturin, Posicor, Vioxx. All these are drugs that have entered the market after being tested in the mice as safe and effective, but were withdrawn from the market. They were taken from the market away due to lethal consequences to humans. Some of them caused cardiac, some of them caused bladder issues, and so on. So they were withdrawn from the market. So what happened? They spent $1 billion for 10 years discovering and marketing these drugs, and they took it out. But they were all approved based on the animal studies. So these are after getting the market. Many of them don't enter the market because they fail in the clinical trials on the humans after showing positive response in the animals. So all this put together, what I'm trying to tell from this slide is the animal model may not completely respond for all diseases. Having said that, I would like to tell that animal models are still useful for understanding the disease, for understanding the pathophysiology, the mechanism, the infectivity, or the response. But we should not completely rely on animal-based studies to create a drug for humans. So that is the concept that you want to understand first. And now I can talk about what we did to address this problem of animal in research for drug discovery and how we are going to move forward. So this is where we created cell culture cell lines from the human cells. If you look at human cells, we have cell lines that are available as cancer cell lines from ATC or many Lonza or any company, like even within India from the cell culture ICTs and so on. So we can develop and we can source the human cells, but there are drawbacks to it, which we'll understand as we proceed. Here are colored images of spherical balls of cells. The left one, that yellow orange, is the fat cell from the human that has become a fat globule or a fat spheroid. The next one in blue is the model that we developed for COVID, came from the lungs, and it's actually stained, DAPI stained, so you can see that. And the third one is a familiar rhodamine stained section for the cell membrane, outer membrane. So membrane is stained reddish fluorescent. The next one is a mixture of DAPI and that both. And the bottom brown clay-like one is the hepatocyte or liver spheroid. So all these are spherical structures present in each well. So this is the model, and they express different colors based on the stains. And so that we have used different cells from different cancers. All these cell lines, prostate, glioblastoma, colon, colorectal adenocarcinoma, KECO2, which is usually used in cell permeability assessments, lung cells, tumorigenic epithelial cells from the lung, BAS, 2B, A549 have been heavily used for SARS-CoV-2 understanding. I had done experiments using them for SARS-CoV-2, and hepatocytes, human hepatocellular and kidney cell lines. So all these things convey that we can develop colorful spheroids. So in nature, when we develop spheroids, the top line of figures are the way they look like under the microscope. But based on the color reaction that we can look at a different stain or an antibody or immunohistochemistry or fluorescent technique, we can develop different colors for different biomarkers. So we can analyze the images for that. So if we know that these are called biomarkers, like actin, myosin, or fat, or in the hepatocytes, glycogen, anything like that, then these become biomarkers. You can also use cytokines as biomarkers, cell culture, cell joint junctions complexes like cadherin as a biomarker. So based on those, now we understand that we can develop spherical structures. We can stain them and get an output of color. Now, based on the intensity of the color or the expression of the color or no expression of color, we can tell that, okay, something has gone wrong. So thereby we can measure the efficacy, toxicology, and everything. So basically what I'm trying to tell is it's better than monolayer because it's a 3D structure. 3D structure is round spherical. In the human body, all the structures are 3D. Nothing is growing on a glass surface, only exposing one surface, top surface to the reaction, right? So spherical cells have advantage, spherical spheroids have advantages. They behave like humans. Are you all with me?
Yes, sir.
Hello?
Yes, sir.
Yes, sir. Yes, sir.
Okay.
Yes, sir. Please
Okay.
continue.
Okay, so that is how we develop these spheroid spherical structures, which are 3D structures. And this is an easy thing. This is easy to do. These experiments are not challenging. And when we look at pictorially what happens, when you bring the cells together, multiple cells, they try to adhere in different directions on different surfaces. So multiplanar, so 3D. On a flat surface, it's only side-by-side attachment. One cell attached to the other one, the other one to the other one, like that. So that's not the right thing. And the bottom is attached to the glass, so you're not even exposing the bottom to the reagents of the experiment. So this is definitely a better model than monolayer, but better model than animal because we are using human cells. So two big advantages using spherical models called spheroids. Why the cells aggregate? Because you all know there is extracellular matrix the cells secrete, like collagen, integrins, laminins, and so on. And they try to bring the cells together. And when the cells come closer, they express some other biomarkers or cell junctional complexes called cadherins, tight junctions, and the cells get attached. When cell matrix brings them together, cells attach together, they tend to form a spherical structure, and that is how a cell aggregation leads to a spheroid formation to the right side. So there are three stages. One is interaction, second is expression and accumulation, and third one is attachment. So here is a picture of a spheroid that I stained with the epi for you to see. And when we put it the most important part of doing this assays is the imaging. So imaging is very important. We can actually rotate the image here. And as the image rotates, you can see the spherical nature of this whole globule that developed. So we can look at multiple directions. On a confocal microscope, we can do this quite easily. And we have a confocal microscope that actually looks at the entire surface of the spheroid. Not only that, the confocal microscope also can go and slice the spheroid, imaginary slices based on the penetration of the light. The round structure, you can see what is at the top, you can see what is at the 2/3, 3/3, center, again the bottom 3/3, 2/3, and the bottom. So we can divide a 500 micron spheroid into 4 micron slices, and each slice you can look at the cells or layers. So from the top, it's like slicing a fruit. You can look at top surface, just the skin, bottom surface, some flesh, another, in the middle you get the seeds, and then go. So you can look at the whole center, and based on the different staining patterns, you can look at, are the fibroblasts more, are the other cells more, hepatocytes more? Did the virus kill the hepatocytes? Did the virus kill something? And you can understand many things. Did the toxin affect the particular cell? So you look at the whole spheroid at different levels. Did it affect the surface cells or the deeper cells? So now you get an output of data. I'm hearing some voices, but fine. So we can see a lot of these cells are now characterized. So now I'm introducing to you a word spheroid, which you all understand now. It's a spherical ball, which is on the right side of the picture. And then I'm also introducing a particular term called organoid, which probably you all heard. But organoid is nothing but its direct representation of a particular organ, because these cells come from primary cells of the organ. Like an intestine or a lung would form something organoid that's like this, with cells expressing their characteristics, like columnar cell or a flat squamous cell. This could be a lung, and then a gut, and like that. So now there's a direct representation. What's the difference between the two? Let us look at it. In spheroids, the cells are coming from cell lines, like cancer cell lines that you can purchase. These can be mixed together and made into multiple mixtures with other tissue cells. So that's a spheroid. In organoid, you're actually picking one or two cells or some few cells that are called stem cells from the organ itself, like from the lung with a lung lavage, where you get the fluid out of the lung. There could be some stem cells, and that can develop into a lung alveolar structure. So that is an organoid. So now you are using stem cells or pluripotent cells or cells that can grow on their own into becoming an organoid that is coming from the primary organ itself. And the primary organ here could be a tumor also. That is an abnormal thing, but it's a tumor. In a 3D assembly, what the spheroid looks like, they self-assemble, involve cell-cell aggregation that we talked about, extracellular matrix, cadherin. They come together and form spherical ball. In the organoid, the cell organization and assembly involves cells responding to physical chemical cues from the organ itself, because organs also secrete growth factors and their own factors. So stem cells secrete a lot of growth factors. So organoid is a better complex structure compared to a spheroid, because spheroid is uniform, it's from some person, some source, and it's very uniform. In an organoid, it could be from different people, and it could be from, it represents much more heterogeneity. Now, the third difference between the two is organ physiology. The layers of heterogeneous cells, quiescent, transient, resemble 3D cellular organization. But in an organoid culture, there could be different cell lineages. For instance, if you pick the lung, there could be alveolar cells, there could be macrophages, there could be epithelial cells, type 1, type 2, there could be blood vessel cells. So all of this put together can also perform an organ-like function. So this is an organoid. Organoid is a development over spheroid, much more expensive than spheroids, but gives much more information that is relevant. So organoids are difficult to culture. They require a cocktail of growth factors. So it's expensive. And it's also but from the sourcing of the cells is from a particular human being. So sourcing is also difficult because you need approvals, you need a patient or a person, and cultured spheroid cells are just sourced from commercial. So that is the biggest difference is spheroids are first things. Development over spheroid is an organoid because it adds more value and information. So organoids can come from terminal differentiated cells, which could be coming from a particular tissue like a stomach or a colon or a small intestine. You isolate the cells from that organ, bring them together, purify, put them in the wells and expand, and then they form organoids. Okay, that is important. That is one source directly from the organ. The second source is you isolate the stem cells, do some genetic engineering or not. You put them in the wells, they aggregate, and those embryonic bodies, you know the three layers, right? Endoderm, mesoderm, ectoderm. And those layers can be again converted into origin tissues. What does a mesoderm give rise to? What connective tissue, right? What does an endoderm give rise to? Blood vessel. What does an ectoderm give to? Skin cell. So you can develop different organs sourcing stem cells, but you need a lot more growth factors, trigger points, timing, much more complex. But you eventually get expanded organoids in two ways. One, directly from the tissues that have stem cells. Second, isolated stem cells, which you manipulate and convert them into a cell type of your need. For instance, you want neuronal brain organoid, you probably go to endoderm, ectoderm, mesoderm, and then isolate the cells and develop this. If you want fibroblast collagen, then you go to mesoderm and get the connective tissue fibroblast, so on. So you have to stimulate with particular growth factors, which we'll talk later. So this is what happens. So the organoid spheroid, I looked at ways to form, right? You're thinking about, do I just drop it into the well and they form? Or do I give them support so they attach? So there are multiple ways. The first one is you give them a matrix scaffold, which is a physical structure like a matrigel. So I explained it in the description. The stem cells are placed in matrigel. And based on the matrigel, they form the spherical structure based on the shape of the scaffold that you put. So it can form a spherical scaffold, you can form a rectangular scaffold. You can do that and populate it with the cells. Or you can do it with centrifugation, where you spin it and then the cells go to the bottom and then form a spherical ball. Or just use hanging drop, where you put a drop of the medium, put some cells, and turn the plate upside down. So now the drop is hanging from just like water drop from the top of a glass plate or something. And in that drop with the cells tend to form a round ball. Because of simple gravity and suspension, they form a spherical ball. That's called a hanging drop. Or you can attach a plate which is not adherent. So the cells don't attach to cells, they're freely moving. And then they form spherical balls. So I use that method to form my spheroids. Those are called ultra low attachment plates. And then you can also do magnetic levitation, where you push the cells from the bottom with magnets underneath the plate. So the cells repel and then go up and down, up and down. So thereby they are suspended. Or you can bioprint, which is 3D printing of the cells itself, where with the needle, the cells can be dropped one by one at a particular speed and they form a spherical structure. And the needle can move around, come up slowly, and as they top it, they form a spherical structure or a structure of any shape that you want. And you have micro patterning and microfluidics, which is you can move the fluids through them and so on. There are many advantages and disadvantages of doing most of these things. There are costs associated with some of them, and it's also simple or moderate or extremely difficult. So obviously hanging drop and low adherent plate methods are the simplest ones, and the complicated ones are the scaffold, centrifuging, and 3D printing. And I use all of them for different reasons. So I will explain them to you later. So now, just to summarize, we wanted to replace or reduce animals because animals are not the best models for some diseases. We want to use human relevant spheroid structures, spherical structures. They are better than monolayers. Now we said spheroids are easy. We went and said we source stem cells or cells from the organs and form spherical structures called organoids. That is a development over a spheroid. So it's become very complex now, right? So let me just stop there for briefly and talk about one disease, but I would like to know if everybody is following it at this point.
Yes, sir.
Okay, thank you. So when I
Just one
said,
question, sir.
yeah.
How do you ensure this blood vessels in the organoid?
Okay, that I'm going to talk about that actually when we talk about tumoroids and organoids. So there are two things. Where the way we have addressed that in our lab is to create tubular matrix structures within the scaffold. When you have a scaffold that is like this irregular, we put tubular structures inside that are also transparent structures that can let the material pass through. But we put, when I said tube, there's not this big tube. These are 5 micron, 6 micron tubular structures. We insert them or put them inside or create those plugs. Then in those particular things, we just put HUVEC cells, human venous endothelial cells. So that's a cell line. So that will just grow in the tube around like a blood vessel. By nature, they're just attached to the surface and they form a ring-like flat endothelium. So that becomes a blood vessel. And you can form a pattern of your choice based on the tube. So you can create a wavy tube or a loop or tube or straight tube. You can insert in the matrix, and those tubes you inject with the HUVEC cells. The rest of the matrix you inject with the cell of your experiment, hepatocyte or intestinal cell or stem cell for that wants to become brain organoid, anything. So now that is the complicated, good question, complicated aspect because most of the times organoids without blood vessels, hard to understand the transport mechanisms and so on. So I would suggest that you follow this method. If not, there are other methods of mixing the endothelial cells at a later point after the matrix is formed, after drilling a hole in the matrix, then putting, so you can also do that. So multiple methods, it's not easy, so it's complicated. Anyway, let's move. Why are we using these spheroids, organoids? Because we want to create disease models. So in our lab, we created a disease model for a disease called NASH, which is also called MASH now, metabolic associated steatohepatitis, also famously called as non-alcoholic steatohepatitis. Many of you probably know this. This is a very, very prevalent condition, and 40% of people in India or rest of the world also probably have fatty liver. So if you look at healthy liver on the top, the next one is NAFLD, that is non-alcoholic. So you don't have to drink alcohol to get this. Anybody can get it. This is overconsumption, obesity, lack of poor metabolism, and all these things. So NAFLD is non-alcoholic fatty liver disease. The liver accumulates more fat. That's reversible. At that point, it's also reversible. With more fat, you have a triggering of inflammation that attracts inflammatory cells and fibroblasts. So that creates inflammation. And from inflammation to the next one, cirrhosis, which is fibrosis of liver, it becomes irreversible. So NASH is inflammation. NAFLD is fat accumulation. Cirrhosis is fibrosis. So 40% have NAFLD. 25% of NAFLD people have NASH. At this point also it is reversible. But 2% out of NASH becomes cirrhotic, which is irreversible liver damage, where you are looking at liver transplantation and so on. And 2% of the cirrhosis may also become cancers, tumors of liver, hepatocellular carcinoma. Okay. So if you look at the histology at the bottom, the second row, you have hepatocyte, nucleus. Now you have fat in the NAFLD, fat droplets are accumulating. Now we have fibroblast inflammation, so collagen is coming into play. Now inflammation leading to replacement of hepatocytes, now that leads to scarring or fibrosis. And now you have dead cells on top of it. So this is a disease, and the reasons for this disease to happen is obesity, diabetes, cholesterol, high BP, and many people have at least one of them in India, right? So anybody can have this. So 40% prevalence. So now there is no good animal model for this disease, believe me or not. There are animal models that are expensive. You give the rat high fat diet that is deficient in an amino acid called choline, they develop fatty liver in 3 months. So that is a model. But then it's not human. It's not developing the way humans develop it. It's induced in a different way by high fat. But humans could be It's high fat, inflammation, many, many reasons. So not the best model. And many drugs were discovered using those animal models. None of them worked. None of them succeeded. NASH is treatable by controlling obesity, diabetes. So any drug that affects obesity or diabetes may indirectly be beneficial for treating diabetes. So one drug from Zydus from India is against diabetes and obesity is effective for NASH as well. So there is no direct treatment of NASH, but targeting obesity or diabetes could treat NASH. Let me tell you what are the 5 drugs that are available that I tested, 4 drugs on the NASH model that we created. How did I create the NASH model? I took hepatocytes, took stellate cells, Kupffer cells, endothelial cells, fibroblasts, everything that is present in the liver. And it's a complex thing. You don't put it as a mixture in one glass or one cup like this and say form NASH. No, it doesn't happen like that. What we did was we grow the hepatocytes for some time. Then we add palmitic acid or oleate. Then it becomes fatty. Then we put some Langerhans and stellate cells. Then it creates smooth muscle, myofibroblasts, perivascular. Put endothelial cells, becomes endothelial. So it's a phased manner. In 5 days to 15 days, you develop a hepatocyte model or a liver model and then induce it with fat. So it becomes fatty. Induce it with inflammation, it becomes NASH. And put more fibroblasts, it becomes cirrhosis. So like that, we developed the model. So there's no animal model. And it took, instead of spending 6 lakhs or 8 lakh rupees, you spend 1 lakh to get this model. And I tested the drugs that are available in the market, like selenosertib, pirfenidone, elafibranol, ALK5, and all of them put together, have been experimented on the NASH model here, showed that what are we looking for? We are looking for amount of staining for fat, collagen, and smooth muscle actin. So when we look at all these things, then we have a response based on intensity of light. ALK5 is a better drug compared to elafibranor, pirfenidone, or selenosertib. And that's exactly what the results even in animal studies showed. So spheroid also, the NASH model also showed the same thing in vitro at a fraction of its cost. But none of these drugs are unfortunately effective for NASH in humans. Pirfenidone is an approved drug for lung fibrosis, but it does not treat liver fibrosis. So this is how we evaluated proof of concept that these drugs could be tested effectively at a cheaper price in an animal model. Like that, we created, this is a pictorial representation of a lung organoid, organ on chip, for a lung that we developed in our lab. And the development of 3D air, because lung is a little bit more complex than liver, right? Lung has to interact with air. Liver has to just interact with blood. So we had to create surface air interaction. So we had a cross section of air also present in the system. And pictorially, the way you want to look at it is, this is a chronic kidney disease model, CKD, because we created liver fibrosis. We took the drug pirfenidone, ALK1, and elafibranol. All these are fibrosis reduction drugs. Now, kidney organoids with fibrosis are in these wells. You put these drugs and you measure the amount of fibrosis by looking for collagen staining. Amount of collagen secreted is directly proportional to intensity of fibrosis. So if it goes down, the fibrosis is getting treated. Simple model. Like that, chronic obstructive pulmonary disease model, COPD, where we tested beclomethasone, betamethasone, budesonide, and NSE, N-acetylcysteine. So usually in humans, they are taking it as a inhaler or as a syringe or an incubator into the nasal, right? So now you put them onto your lung fibrosis model here, and not the fibrosis, but the mucus reduction model, the goblet cells, and you measure how much mucus is secreted from the wells. And if the drug is effective, the mucus secretion is less. So like that, we measured. So we can use it, this pictorial, but we are developing these models. We have to source for cells from the kidney, right? So we go to the patient and get kidney biopsy and get direct source of kidney cells. For the lung, we get it from the direct bronchial lavage from the lung tissue, and so on. What is the workflow? Go to the patient, get the tissue, slice the tissue, prepare an organoid. Now, I understand that you know there are multiple steps here. Grow them in the wells or the growth plates, stain them, image analysis, drug treatment, image analysis, analysis of the imaging data, and interpretation. Now you build this data over a long period of time for multiple patients. Now you have an AI model. Just look at, give the result, and it tells exactly the intensity of fibrosis, the effectiveness of the drug, and so on. So this is what we try to do in our lab. This can also be done by ex vivo. You don't have to build an organoid or anything. You can also source the tissue directly from the patient, slice it, put it in the well, and also do it. But the growth of those cells without other support systems is very minimal. So it's very different. So we do use this system to develop organoids. And the readouts usually are, like I said, is a colored image for a specific biomarker, or it could be supernatant in the well that secretes a particular cytokine and use it in the imaging, the Z stacking. Again, I told you about multiple layers of the cells that get affected in the image. So these are the ways to measure them. Summarizing the organotyping, you have spheroids. Now talking about multicellular spheroids, multicellular tumor spheroids from the tumors, and tissue-derived spheroids, which multiple cells. Okay, we'll not talk more about this. Let me go to the next thing. So now we talked about monolayer, spheroid, organoid, of course, blood vessel integration, testing of the drug. Now we talk about something is missing. All these are looking static, right? In the well, you put a ball of cells, it is static. And it is static in a fluid. In the real life, nothing is static. Cells are static, but there's a constant flow of blood, constant arrangement for them to interact and exchange fluids. So we created microphysiological systems, also called as MPS. The most familiar term for this is organ on a chip. So if you look at this, we get the organs from different regions of the human body and then put it into one single slide or a deck, which is probably 10 millimeters or 4 millimeters thick, and then like a slide, but it has multiple things. And with a perfusion or a fluid or a motor or gravity, you can use the flow pattern. So cells get touched by fluid. So let's say regular pattern, gut, then it interacts with the liver, then it gets absorbed, goes, touches the brain, heart, comes back, touches the kidney, gets excreted. So you can create that flow artificially. So that's what we try to do. And we have done it with human organs from different organs, trying to create an MPS system. You can source them from one patient. All these organs, of course, a cadaver, a freshly dead person's tissues, or a volunteer who wants to give his tumor tissue or something like that. Like that, we can develop organoids from different organs, put them in a microphysiological plate at different points, and ask them to look at. And if you look at it, I also listed the different growth factors that we need to put for those organoids too. So in each well, if you have liver organoid, you need to put all these growth factors at different concentrations. So it's complex methods, but these organoids, if you look at it, are different. Some are common, like noggin is a very common growth factor that is used for almost all of them. But then you have to use specific growth factors for converting these potential stem cells into particular organoids. The advantage of that is the biomaterial, the matrix, the cells, the organ, the systems tend to talk to each other. Now you have a humanized, not a human, but a humanized system with all the organs on a particular dish called the chip. It's the human on the chip or multi-organic system, and you can use it for testing drugs. And then if it's coming from one person, you can probably personalize the drug treatment based on the response of that particular person and advise on that. So that could be an application. So look at the model here, a little bit more depth. You have the culture disc flask. You have pattern here. You insert what is needed, other organs. You grow it into an organoid. You create a microfluidic chamber where you can come in kidney, liver, gut, brain, and the fluid goes in between all these four. So you have all these things. Measure the cytokines, measure the organs by histologically imaging, and you get a complete picture of how it is. So the difference between this and a simple organoid is it's fluidic, it's dynamic, and it's a complex in vitro model. And FDA recently approved 2 drugs, one for cancer and one for neural, based on this output of results from this kind of MPS system. Because so obviously It's very important for us to understand that MPS is much more developed or improved over spheroid and organoid. Now I switch the gears and go to how I use my spheroids and organoids for tumoroids, which is nothing but cells coming from the tumor. Tumor is cancer, and I make it into a malignant tumor spheroid. So it's as simple as that. So we call it as tumoroid. The advantage is, let's say we obtain cells from patient 1 on this one picture, and then there's patient 4, another picture. You look at, there is a difference. The green on this picture from patient 4, the years of survival is less than 1. This person is dying within 1 year. So the cells look pleomorphic, which means different sizes. The matrix is very loose. They're behaving very wild, and the matrix is actually coming out. So a lot more secretion coming from the cancer cells. And then here, this much more circumscribed, more organized, not looking more lethal. So this person survived for 3 years. So based on the growth and morphology of the spheroid that you grow from the tumor tissue, biopsy from the patient in the lab, looking at the characteristics, you can tell prognosis. You can test the drugs on this and tell which drug is available or which drug is testable. Another dimension is when you source the tumor cells from this and form an organoid, you can also do genetic sequencing, NGS. You can look at the genes expression for cancer. From the organoid culture, you can look at drug testing. Both combined, now you have what drug is effective based on what gene is present in this person. So you can find an effective drug. You can actually create a bank of organoids. Biobanking is a very popular thing now. And we are using this biobanking. You can screen different drugs and use it for drug discovery. With what particular kind of morphology, you can pick that biobank of tissues and look for what drug is effective with if the person has this morphology of the behavior of the tumor. So this personalized medicine. So if you actually put the tumor into the mouse that can accept human cells, you can create an immune system within the mouse. We call this as humanized mice. So you took a mouse, you put the cells from the tumor of the patient into the mouse. The 3D culture is there. Now the patient tumor is carried by the mouse. But this mouse is very special. This expresses human immune cells. These are special mice, immune compromised. I'll not go deeper into that. So here there is a marriage of in vitro cells, tumoroids, with the in vivo human, which means the microphysiological system is addressed. But then you can understand immunology because this mouse accepts human immune cells. So then you understand how the immune cells are reacting. These are called humanized mice. So a combination of tumoroid, humanized mouse gives a lot more information for cancer research. So just summarizing, get the tumor tissue, make a spheroid, inject into the mouse. That mouse has the immune system, do the molecular profiling, test different drugs, and then you have clinical development of particular drug. So this is important for drug development, understanding the cancer, and then using AI/ML for doing much more research. Patient-derived tumor xenografting, this is what it's called. It's very popular. It's been extensively used, continues to be used. I use it quite a bit being a veterinarian. I like it because it marries both human cells, organoids, and the microphysiological system of the mouse with the acceptance of the mouse of human immune cells. So this becomes a beautiful system for measuring. And I have done NK cells, CAR T cells, CRISPR-Cas9 genome, all these things on this model. And now we have results available for various cancers using this model. I have not gone too deep into this. You can screen many compounds. You get many results. You have combinations of compounds. You can do systemic capsules, oral, dermal applications, and all these things. And you have curves for immunotherapy, what immune response you are seeing. The weakness, of course, is the cost. Over time, the human stroma is replaced by mouse stroma, so it's difficult to keep the humanized mouse for long. So you have to do the experiment within 6 weeks. So it's a temporary window period where you have to do all your experiments. But the beautiful thing is you can look at the tumor patients, organoid models, look at the drug response by genetic as well as response immunologically, and select the therapy of choice for the patient. So you can go back and tell the oncologist what drug or combination is effective. So this is one of the better ways of doing this. So bottom line, is you have patient tissues, which is important, organoid, which is important, uh, bioimaging, cytokine measurements, biomarkers, uh, genetic profiling, all important. Then you have drug treatment and further analysis and interpretation by AI. Putting all of this together, you have a marriage of advanced models called 3D models with animal cells, with a mouse, and then that leads to effective drug design and everything. So a mouse may, by itself, may not be a good model, but a marriage of all these things makes it a better model. So you reduce the number of animals. But even without this, you can do it in vitro on a microphysiological system and still do a lot more experiments. So the advantage is in vitro, it mimics the tumor and translates the preclinical into clinical success. What is a forward strategy for us? Not just discovering drugs, finding drugs, testing drugs. The forward strategy is to find personalized treatments. A person comes to an oncologist with a particular cancer. The oncologist can take the tumor from the person, give it to the lab, and we can develop a tumoroid. Now you can do the genetic analysis, test the drugs that the oncologist wants to use, even experimental drugs, and tell the oncologist which drug was effective. Within 30 days, you can do this experiment. And now the oncologist knows exactly what drug to give to the patient. So we are using organoid, tumoroid, microphysiological systems combined with genetic markers, biomarkers, imaging, and AI to tell the oncologist what is a good drug. So this is the impact of this technology, not just discovering drugs, but also therapeutic treatment of the patient. So with that, I will end my talk on the 3D models, which included spheroids, organoids, microphysiological systems, patient-derived tumors called as tumoroids, marriage of all this with animals, which becomes PDX models, and we can use it for drug discovery as well as for treatment. I will stop sharing, and if you have questions, please go ahead. I hope you all understood this presentation.
Thank you, sir. Such a informative and enriching session. And I now request all the participants to ask the question in the chat box.
I'm seeing some questions here. Yes. Yeah, it's an
Yes.
alternative model to an animal model. That is very well said. Yeah, it's alternatives are getting very popular. The FDA Modernization Act says that you have to cut down the use of animals. Of course, you cannot escape it completely because there are no alternative models for many diseases right now. There are rare diseases that does not have animal models, so alternative model is the only one that you can use, and so on. So you have to be you have to expect that in the next 5 years there will be a lot more alternative models that will become acceptable. Any questions, please?
The one question I have, like when we're talking about liver model that you added Kapha cells, that after some days you added some other cells. So how do you ensure that the composition of your organoid is similar to what actually liver has? The percentage of different types of cells.
Yeah, so
It may happen that more Kapha cells are there in your organoid as compared to what actually liver has.
yeah, so it
So how
is
do you
very
maintain
similar.
this?
Very similar of a typical healthy liver is what we used as an Excel. So there's an ex vivo liver that we took, compared it with this spheroid or hepatocyte spheroid or liver spheroid that we developed. And the proportion of the cells have been figured out by biomarker staining in both of them. A normal slice of liver and a hepatocyte spheroid slice liver. We looked at the different biomarkers and it's very similar. But if you don't add the specific proportions, you will have differences. And every person's liver is different. So that heterogeneity is good to have actually. Only thing is you need to have statistically different, like we have a 96 well plate with 12 different spheroids of the liver. So every thing will not be the same. It will be similar, but not the same. That's why the statistics is important.
Also, sir, one more, like how far this, first I wanted to know how do you make this mouse with human immune system? And secondly, how far has this technology, what is the status of this technology in Indian scenario? Like
Very good.
how,
Okay,
yeah.
these mice are available in India, by the way. I was very instrumental in bringing these mice to India. So these mice are called special immune compromised mice. It's called NCG, NOG, NSG, based on who the vendor is. When I say these are immune compromised, these mice lack secretion of antibody by B cells. They cannot secrete T cells. Some of them lack NK cell activity, natural killer cell activity. So there is no graft versus host rejection also based on the MSC compatibility. So these are mice that are genetically modified by knocking out the abilities to release B cells, T cells, macrophages, and NK cells. So they are immune compromised, number one. Number two, they also are irradiated with radiation, so they don't really respond when a foreign thing enters their body. So they're immune compromised, further immune compromised. Now, when you inject the CD34 cells from the bone marrow of a human, CD34 cells, they get populated in these mice as human immune cells because they are not rejected, they proliferate. Now, if you take tumor from the human, then the human tumor and the human immune cells in the mouse are interacting. So, creating the mouse itself is a challenge, but these mice are commercially available, expensive, but available. And that's how it is. Now, the recent advances, you don't even need to do irradiation. You can use the mouse directly without an irradiation for them to accept these cells. But like I said, they will become leaky mice after some time. So you have to be careful with that. Yeah. You're on mute.
There is, yes, sir. So there is one question in the chat box. Will this 3D
I'm
model,
saying it. Yeah.
yes, yeah.
Will this 3D model reflect the same nephrotoxicity conditions as mice model when testing drug nephrotoxicity? Okay. Uh, Good question. Mouse has a mouse kidney, number one. The 3D model here has a human kidney. So big thing. But then in a nephrotoxicity model, your toxicity response for a human nephron, glomerulus, mesangial cells, and everything, you want to know the toxicity for the human nephron, not a mouse nephron. When the mouse nephron is affected, you expect the human nephron to be affected as well. That is when you actually test it for in the spheroid and see if it is having an effect. Of course, yes, nephrotoxicity, before putting it to the mouse, I would advise you to take a monolayer of kidney cells and see if it is toxic to those kidney cells. Or you can take a 3D model and then look at the toxicity of the 3D model of the kidney. So it's effective, and nephrotoxicity is a good question. But again, this is an early time. There are challenges of doing this. I would say toxicity depends on multiple things, right? It depends on loss of excretory ability, thereby accumulation happens. Lack of modification of the toxin, so the accumulation is toxic. So I would go with a mouse model for nephrotoxicity because it encompasses the physiology. Just particularly on a kidney cell line, if you want to see the toxicity, then the 3D model is better. Both are reflective. So good question. Both are reflective, but you have to know what is the toxin you are testing.
There's one more question, sir. Dr.
How
Saroj has
economical it is for developing. Spheroids are cheap. Organoids are cheaper than microphysiological. Microphysiological systems are a little bit more expensive, but all of them are much more cheaper than mice and rats and animals. But the biggest challenge here, I think it was Saroj Kumar, who was Dr. Saroj Kumar. So the biggest challenge here is the manpower and the skilled manpower to develop these things. Because it's early phase, it's a lot of, you'll find a lot of lab animal veterinarians, lab animal people injecting something, getting the results out of it, analyzing with HPLC analytics and ELISA and all that easy. So that's a very established system. So it is easy now. So hiring the right people is the most economical challenge here. And the thing is, these models in India are not yet available in many labs. Very few labs doing it, but the world is changing. These models are getting accepted, so there will be, like for instance, 5 years back, only 2 or 3 labs were doing it, like mine, and now there are 60 or 70 labs doing it. So it's a development, and in the world there are many more labs that are. So it's going to happen. 5 to 10 years, you'll have many more. Economic development cost will come down as more people start using it. Growth factors are expensive.
So just a personal question. How much do you charge? How much your company charges for such studies? Like
For
if
a
if
simple molecule for efficacy is how effective it is on a cancer. We test it on 12 cancer types cell lines. We charge 25,000 rupees per molecule. If you want it on a real patient cancer tumor, that costs 2 lakhs. Yeah, it ranges from 25,000 rupees to 2 lakhs for one molecule.
so so does Indian population show interest in such models testing?
We are
What is the
talking, yeah.
status?
So we are talking about contract research organizations, CROs that provide services to foreign companies. So those people want this to be done, and but India, the drug discovery is less, so we are not getting many things. But there are academicians, many people in the universities, they want to test their drugs without spending too much money. So I get a lot of those people, startup companies, academicians who have, sir, I have a herbal extract. I want to see if it cures this cancer. Can you test it on it? And I said, I will test it on 12 cancers, not just one cancer. So that's a good thing. If it's an active ingredient, it's beneficial for them to test it. So yeah, that's where the interest is coming from academicians and startups in India. But the interest from the outside world, outside India, is a lot of drug discovery, major pharma companies willing to do these tests. And few labs in India offer this test. And I'm building a lab for this one. Yeah.
There's some chat questions are there.
Yes, it can be an effective antifungal compound acceptability. It's a very good question because there are a lot of skin 3D models available. Now, if you look at cosmetics, there is no animal study. It's things, drugs, cosmetics get approved just based on 3D skin model assessment. So this is a best example that you can find for in vivo testing, and it is acceptable. That was
And
from
one
Jyoti.
more question is, are you offering project for master students? Dr. Saroj is asking.
We don't offer internships. We offer advisory roles as I become an advisor for their masters and then sponsor if they want to do a small study or experiment. We do it in our lab uh, and we don't charge the students. If it is a compound that is less than 25,000 rupees expense. But anything more than that, we want them to write a grant or provide some resource because it costs for us to do this. So we do help. I do help a lot of students in my lab. And close to 50 or 55 students have used my services for their program, and many of them have gotten postdoc opportunities in USA because this is a very advanced model which is popular in US. So I do help them. I charge them based on the project, or I don't charge them. So the reason why I don't charge them is if there's an experiment I'm doing for a CRO or a academician who is paying, there could be 20 wells that are free on a plate. So I can put few cells in that one and test it. That's cheaper. So I don't charge them. But if they want their entire project to be done specifically in one plate, they need to pay for that. Thank you so much for some of the people who are saying the talk was good. Thank you so much.
Any other questions we have?
Thank you, Shilpa. Thanks for giving me the
Thank you, sir,
opportunity.
for yes, sir, and we are also grateful for accepting our invitation. And we just saw your contact, like Chat GPT suggested us, one of the
Sure,
experts in this
sure. Thank you.
area. So
Thank you so much.
that
I give this
itself
talk
shows
many
that you have
places.
already yes, yeah,
There is a 3D conference coming up in Goa, so I'm giving a talk there as well. Panel. So
yeah, yeah.
thank you so much.
Thank you so much, sir. Thank you.
I'll
Thank
be
you.
in touch. Thank you. Thanks, everyone. Have a wonderful evening. Bye.
So we'll post the link for attendance and feedback in just 2 minutes. And the next session will be at 7:30. Till then, we can have a small break. The meeting is on and we can rejoin at 7:30.
Thank you.
Thank you, sir. I'll just post the attendance link. Hi, Vikas. Dr. Vikas, how are you? Can
Hello,
you
Dr.
hear
Shilpa.
me?
I'm good. How are you doing? Can you hear me?
Yes, yes. I'm so glad that you joined at 6 a.m. in US, and
Uh,
he has
yeah,
been
no
so
worries.
kind. He's so kind that he's giving early morning talk from US, but for us it is our leisure evening. So Dharmesh, I request you to make him co-host. Just let me see if you are. Dharmesh, can you make him co-host? Now you're co-host. You can share your screen. So we welcome Dr. Vikas. I request Twinkle to introduce our speaker.
Can you guys see my screen just to
Yes,
begin with?
sir.
Yeah,
Okay,
yeah. Yes, sir.
great.
Thank you, ma'am, for giving me
Twinkle,
the
are you there?
opportunity.
Oh, yeah.
Yes, ma'am. Am I audible?
Yes, yes.
Okay. Thank you, ma'am, for giving me the opportunity to introduce our esteemed speaker, Dr. Vikas Bhatt. He is a director of CMC Strategy and Program Leadership at Biomarine Pharmaceuticals, with over a decade of experience advancing biologics, gene therapies, and oligonucleotide programs from early development through commercialization. He leads cross-functional CMC team spanning formulation, analytic development, manufacturing, and regulatory strategy, and has supported multiple successful IND, CTA, and BLA submission globally. Dr. Bhatt earned his PhD in biochemistry and molecular biology from the University of Miami and completed postdoctoral training in protein biochemistry at the Scrip Research Institute. He holds a BTech in biotechnology from Guru Gobind Singh Interuniversity University, providing a strong academic foundation that spans biotechnology, molecular biology, and protein science. This research to industry training underpins his deep expertise in biologics characterization, viral vector analytics, and formulation science. He has authored more than 50 peer-reviewed publications and patents and is recognized for translating rigorous science into scalable, phase-appropriate CMC strategies that accelerate clinical and commercialized success. Now I request our esteemed speaker to kindly proceed with the session. Thank you.
Thank you so much, Twinkle. Can I go ahead, Dr. Shilpa?
Yes, please. We are eager to
Okay.
listen to your talk.
All right, sounds good. Thank you so much. And good evening to everybody. As Dr. Shilpa said, this is kind of morning time on the west coast of United States and 6 a.m. in the morning. And I'm feeling very energized and enthusiastic about this talk at this hour on this coast. So thank you so much for joining. Really appreciate. And I do want to appreciate the organizing committee to give me this opportunity to present on this topic today. So please let me know in between if I'm going too fast or you have any questions. And also, in case the technology doesn't work out, which sometimes does happen, my slides are not moving forward, please let me know. So what I want to do through this presentation is to explore how the innovation across the gene therapy, RNA, cell-based therapies, and delivery systems is reshaping the treatment of genetic diseases. And I'll focus not only on the scientific breakthroughs, but also on what it really takes to translate these innovations into approved scalable therapies that reach patients globally. As Twinkle mentioned in my through my bio, my overall career has seen the transition from academic to industry, and one of the motivations for me to do that was to see how the scientific breakthroughs that we do as a scientist or bench scientist can get translated for the eventual treatment of the diseases. So this is my disclaimer slide before I start, and I just want to highlight that the views that will be expressed today are my own and are intended for educational purposes only. They do not represent the position of my employer or any past affiliated organizations. And this discussion should not be interpreted as regulatory, medical, or investment advice. Also, another disclaimer is, and as you will see in the rest of the presentation, I will be talking about a lot of modalities that are treated for genetic diseases. I personally am not expert in all of them, so I may become at some point limited in my knowledge in case you have questions. But I'm sure I can take those questions and get back to you through and do some investigation on my part and get back to you after this talk as well. Okay, so why this topic matters, and just to kind of give you an idea that genetic diseases, they sit at the intersection of science, technology, and urgent patient need. Currently, there are more than 7,000 rare genetic diseases which affect over 300 million people worldwide, and yet fewer than 5% have approved therapies. And despite this unprecedented advances in biology and engineering, the vast majority of patients still have no disease-modifying options. And this gap is why innovation in therapeutic modalities is just not an optional, it's not an option, it just becomes that much essential. And just to uh, put my message out there through numbers, as you can see, as I mentioned earlier, these numbers highlight the magnitude of the challenge. We know thousands of genetic diseases, and as we continue to do more research, more of these genetic diseases are coming out to the into the picture, but only a small fraction have approved treatments. Many of these conditions affect small patient populations, creating scientific, regulatory, and commercial hurdles. And this is precisely where platform technologies like gene therapy, RNA, and cell-based approaches become powerful, because they allow us to address multiple diseases using shared development frameworks. So two things that I would like you to remember from this slide is the gap that we have currently in how many genetic diseases are known, and how only like around 5% of these have approved treatments. And the other part is the need for a platform technology that can allow us, which can act as a plug and play for the treatment of many of these genetic diseases. And these platforms can be gene therapy, RNA based, or cell based approaches, which we will which I will go through in my later slides. So before moving further, just to kind of give everybody a sense of what are the different kind of genetic diseases that are known right now. So there are four major categories, and in general, these are not a single category, and hence the kind of treatment which I earlier mentioned needed will also be diverse. So, for example, monogenic disorders are caused by single gene defect and are often well suited for gene therapy or gene editing kind of modalities. Polygenic disorders, on the other hand, involve multiple genes and environmental factors, making them far more complex and not easily treatable through just gene therapy or gene editing process. Chromosomal disorders involve large structural changes, and mitochondrial disorders present unique inheritance and delivery challenges. And understanding the disease biology becomes fundamental to selecting the right therapeutic modality. So once we identify, or once we know what kind of genetic disease a patient has, in general, when we look at how exactly does that impact, or what is the impact of these genetic diseases, that extends far beyond the molecular defects that we just saw in the previous slide. So, it's kind of patients and families often endure years of diagnostic uncertainty, chronic suffering, and significant financial strain eventually. And healthcare systems similarly face high cost, complex care coordination, and limited expertise for rare conditions. Globally, you can think of genetic diseases that contribute to millions of deaths annually, particularly, and this is very important, in infants and children. Some of these problems get worse in developing and underdeveloped countries, where the disease identification, monitoring, and eventual treatment options are very limited. So So in general, what I wanted to do is, through these previous slides, provide some context to the importance of the topic, which we will discuss in the rest of my presentation now. And I would like to review the current therapeutic landscape, then explore emerging innovations across gene editing, RNA, and cell-based therapies. And finally, I'll discuss future trends, including AI, personalized medicine, and regulatory considerations before closing for Q&A. And I hope this builds enough interest in the field, and you can use some of the information presented here in your own work that can help in bridging the gap to treat patients with genetic disorders. Any questions so far?
No, please carry on.
All right. Guys, it's morning time on my side. You guys are on the evening side, so please feel free to be more active and ask me questions. I may not be able to answer all of them because I'm still sleepy. Okay, so starting with the current landscape, or the current therapeutic landscape, when we look at the existing therapeutic landscape, there are several therapeutic modalities which are already in use today. Gene therapy, which aims to correct the disease at its source, but comes with cost, delivery, and durability challenges. Similarly, the enzyme replacement therapies do exist in case where the gene or the protein is missing. So these enzyme therapies can be effective, but require lifelong administration and often fail to reach certain tissues. Small molecules typically modulate symptoms rather than address the root causes. And antisense oligonucleotides, which offer precision but face delivery toxicity and cost limitations. So you can see that, in general, the existing modalities, they all have their own trade-offs, and there is no universal solution. But what this does offer is a lot of area to research and improve for the benefit of the patients. One of the things, I guess we are all, as part of the scientific community, we are all aware of, is that we keep discovering and innovating new treatments or new molecular understanding of the disease or the treatment paths. But this comes with its own pros and cons. So while it does help a lot of patients, it still has limitations, and that's where the scientific community is actively working towards removing those hurdles or the limitations, so that we can continue to evolve these therapeutic modalities such that they can become even more efficient and user friendly, or rather patient friendly. So one good example to kind of highlight the existing therapeutic landscape is that I wanted to share with you guys as case study is the success of the success of these therapeutics before. I'm sorry. The one of these case studies that I wanted to discuss was through the Zolgensma, which is used for spinal muscular atrophy disease, which is a debilitating genetic disease. And this was a landmark example of gene therapy's potential and its challenges. What it does is what this drug is. It's an AAV-based gene therapy drug. It delivers a functional SMN1 gene using the AAV vector in a one-time treatment for infants with spinal muscular atrophy. And this was something very powerful because these kids had, before this, there was nothing for these kids, and these patients or these kids used to die at the age of 2 years, 1 or 2 years, because they were missing this SMN1 gene, and used to go through this muscular atrophy, and within 2 years will die. And Zolgensma proved to be one of the landmark gene therapy medicine that allowed one-time treatment And clinically also, the results were transformative, significantly improving survival and motor function. But it does come at a very high cost. The cost of this medicine at this point is $2.1 million. And this is because of the manufacturing complexity and access constraints, which actually sparked intense debate, and therefore highlights the scientific success alone may not be enough, especially to ensure global availability of these kind of medicines. So again, this is a great example where we know that there was a path-breaking medicine that came into fruition, allowed these kids. Clinically, we showed a really great result, remarkable result from these for this drug. But we know the global outreach of this drug, at least for now, may be limited because of the high price tag that it carries. And therefore, that kind of like creates the gap of what can we do to improve this, to improve the global outreach of a drug like Zolgensma. Okay, so next we move to the gene editing, which has been in news a lot for past years, especially due to famous Nobel Prize Chemistry Award to the inventors Emmanuel Carpenter and Jennifer Doudna in 2020. And in this, we start with CRISPR, which we kind of all have heard about, and especially because of these Nobel Prize winning work, this has become really, really famous and has been applied and is being tested in for the treatment of different diseases. So in general, you can say that the discovery of the use of CRISPR-Cas9, which has fundamentally changed our ability to edit DNA using a guide RNA, Cas9 can introduce precise cuts at defined genomic locations. So cells repair these cuts through either error-prone pathways that disrupt genes or templates-driven repair that can correct mutations. This precision opened entirely new therapeutic possibility, but also introduced new safety and delivery challenges, which I will discuss in my later slides. As we started learning more about CRISPR technology and got better at it, base and prime editing represents next generation refinements of the CRISPR technology itself. So on the left side, it's showing the major, how CRISPR-Cas9 works. And the right side talks about the advanced CRISPR-Cas9 technology, which can be done at a single base, where we can do single base changes, not just the cuts on the DNA, but we can in fact change single bases of the DNA. This has significantly reduced unintended mutations and therefore improved the predictability of this therapeutics, as well as making it not little, but significantly more specific for the treatment of different diseases, where we have identified the location of the genome where we need to make the changes. So in general, these tools expand the range of mutations we can safely address and represent a major step towards more precise genetic medicine. One good example in this case is Casgevy, which is the first and only approved CRISPR-based therapy for the treatment of deadly sickle cell disease and beta thalassemia. Its approval in general, it marked a turning point, demonstrating that gene editing can move from a concept to clinic. At the same time, the boxes on the right side show many other CRISPR-based programs remain in development, including both basic and advanced technologies, which reflect the great momentum and that this field has, or this therapeutic modality has taken for past 5 to 6 years now, or more than that. And also, you can see also that it's not like crazy big number because of a lot of like caution that people are still taking. Hence, there's only one approved commercial product for now. So this slide kind of captures the challenges and the safety concerns, and that this is important I wanted to highlight this because this is where, as a scientific community, we identify the gaps. Like if we are talking more and more about, we talk a lot about the success or the basics of the technology, but also discussing the challenges and the safety concerns, therefore kind of like sparks the need for additional scientific innovations and discoveries that will allow moving the field forward in the right direction. So in general, despite the promises of the CRISPR, it still faces significant challenges in terms of being a mainstream therapeutics for genetic diseases. And therefore, there aren't many CRISPR-based therapies out there in the commercial market right now. The major impacts are the off-target edits, immune response to editing components, and mosaicism. They all pose safety risk for the patients. Or by mosaicism, I mean the incomplete or uneven editing. While these are the technical risk of the challenges of the CRISPR technology, at the same time, the ethical considerations, particularly around the germline editing and equitable access, these are also equally important. And addressing these issues through continuous research efforts hopefully will be critical for responsible clinical adoption of this therapy. So from CRISPR, we're moving on to the RNA-based therapeutics, which is another more recent approach to manage and treat genetic disorders. And RNA-based therapies allow us to modulate gene expression without altering the DNA, unlike CRISPR. This can be used through two ways: siRNA therapies, which silence the disease-causing genes using the cell's natural RNA interference pathway, and messenger RNA therapies that deliver temporary instruction for cells to produce therapeutic proteins. There are multiple approved drugs in both categories, and I'll show that in my next slide. And one really good example for messenger RNA therapy, which we all have seen a live example, is the COVID-19 vaccines, which provided a powerful proof of concept for rapid, scalable messenger RNA deployment, with delivery systems like lipid nanoparticles playing a critical role. In general, I'm hoping that we are all aware of our molecular biology concepts, and therefore I didn't go into the details of what exactly is a siRNA and messenger RNA, but the point here is that these kind of modalities are used at the they modulate the gene expression without impacting the DNA itself, so they work outside the nucleus. Well, not in all cases though. So this slide captures the current overall the therapeutic landscape of this modality. And you can see that RNA therapeutics in last decade have progressed rapidly. We now have approved RNA drugs like Onpattro and globally deployed messenger RNA vaccines, as I just mentioned, for COVID. And of course, the COVID vaccine is not for part of the treatment for the genetic diseases, but I wanted to highlight it because this gives us an idea of like how the proof of concept is there through the widely used treatment option for COVID. The clinical pipeline, as you can see, is extensive with hundreds of programs across multiple phases. And these RNA platforms are increasingly viewed as the modular technologies, rather than just single products. So you can kind of like set up a platform and then use that platform for the treatment of multiple other kind of diseases, not only just genetic, but also you can go beyond genetic diseases towards the say the viral infectious diseases. And just like CRISPR, while this modality is rapidly growing and a lot of disease targets are being explored, RNA therapies do face real limitations due to multiple reasons, and hence a wide gap where future research can be focused at to improve this therapeutic area. RNA is inherently unstable and requires chemical modifications, which hopefully should not take away the therapeutic value. So there is a lot of work that goes into through a lot of genius chemical or chemists who work on modifying the RNA to make it more and more stable, as you guys are all aware how unstable the natural RNA is, to improve its therapeutic value. And the targeted delivery remains a major challenge, particularly beyond the liver. Manufacturing complexity and cost also impact global accessibility, which underscores the importance of CMC innovation, where a lot of work is going into how to make this a scalable therapeutics that can reach to patients across the globe. And therefore, scale up of these therapies is challenging task for now, and making sure it reaches global patient population that does become an uphill battle, at least for now. But the good news is a lot of research is going on in this uh, modality, just to answer all these challenges and hopefully reduce the safety burden for the patients as well. Okay, so moving towards my next favorite topic, which is the cell-based therapies, which is the next frontier of advanced genetic medicines. And one good example, stem cell therapies, which leverage the ability of cells to self-renew and differentiate. These are used in regenerative medicine, disease modeling, and drug discovery. And one of the major advantages of this approach is the ability of induced pluripotent stem cells that allow the patient-specific, which is very important in this category. This allows the patient-specific modeling, which is of huge benefit from a personalized medicine point of view, and may serve as the future of therapeutic sources. But of course, as we have heard this term multiple times, with greater powers, it does bring a lot of complexity as well. So, it's not straightforward, but it does provide that element of personalized medicine for some of the genetic diseases. Another CAR T therapy is a great example of this therapeutic area. You must have heard about it, or some of you might be working on it, or are doing some sort of a research on this. This was originally developed for cancer, and is being adapted for various genetic diseases now. What happens, just at a very high level, the patient T cells are collected, they're engineered, expanded, and then reinfused to target specific disease mechanisms. So, that's where the personalization of this particular modality comes into use, where the patient's own T cells are used for the treatment of that genetic disease for the patient. So, in general, this approach, in general, demonstrates the power and value of this approach by highlighting how cell engineering can serve both therapeutic and the delivery functions. From a clinical progress point of view, in cell-based therapies represent a rapidly expanding field. Dozens of these therapies are already commercialized for the treatment of cancer, and there is a lot of work going on, with hundreds of more in clinical development beyond the treatment of just cancer, and moving towards the genetic diseases. So the number that I'm showing here is kind of a combined number for both genetic diseases as well as the other diseases like cancers. But the point here that I wanted to make is that this is the proof of concept is already out there. We know that this therapy works, and it works at a very personalized level. So, this kind of like in general reflects both strong efficacy signal and the ongoing investment across this sector. Well, From a challenges point of view for the cell-based therapies, while there are many challenges and safety-related risks like other therapeutics in this case as well, which I mentioned earlier, I wanted to highlight the manufacturing challenges, which is the largest bottleneck for this class of therapeutic modality. You can imagine that these type of personalized therapies require complex logistics, specialized facilities, and rigorous quality control. Because, if you can think about it, when it comes to the personalized treatments, you are taking these T cells from the patients, individual patients, then you are turning them around, making them a therapeutic, and then putting them back into the patients. All that needs to happen within a controlled environment with all the compliances and keeping in mind the safety of the patient. And that brings in a lot of logistical hurdles, be it the the experts in the field who can deliver or kind of like work with the T cells. We need to make sure that we have the right kind of facilities where we can kind of use those T cells and convert them into the therapeutics. And then eventually, once all that is done, you have to think of from a manufacturing point of view, from a global outreach point of view, how to scale up that whole process such that we can reach to more and more patients. The turnaround time of the faster we can take the T cells, convert them, and put them back into the patient. All that is like a logistical nightmare that this particular therapy goes through. So in general, when you put these factors together, that drives the cost and limits the scalability, making manufacturing innovation the manufacturing innovation becomes the paramount for this kind of therapy to allow the widespread reach of this particular modality. And keeping in mind also the cost and the efficiency for the final outcome for the patient. Okay, so this kind of highlights my section where I wanted to kind of like give a brief idea or a high level idea of the existing therapies and the challenges associated with them. I do want to touch upon the very niche part of this innovation, which is the novel delivery systems, which allows the safe and efficacious delivery of these target DNA, RNA molecules in the cell. You can think of them as a very futuristic car that carries the biological molecules to their specific address within the cell. Starting with viral vectors that remain central to the delivery for gene therapy. Mainly two types of viral vectors are used in this area. AAV vectors, which offer low immunogenicity and stable expression, but they do have limited cargo capacity. Hence, the size of the DNA or the RNA that we want these AAVs to carry is limited. So it becomes very disease specific. And at the same time, lentiviral vectors can accommodate larger genes and integrate into the genome, but carry insertional mutagenesis risk. So vector selection, because of these pros and cons between the two type of vectors, that becomes it becomes a very challenging task. It is highly indication specific and impacts safety. That can impact eventually the safety, durability, and manufacturability. So, choosing the right vector for the right kind of passenger, if I can say, which can be anything, the gene or the RNA for the treatment of disease becomes really important. It's like picking the right partner who can help you move forward in the life and achieve your respective goals without impacting the overall, safety, durability, and creating cost burden. So moving on to the non-viral delivery systems, which offer attractive alternatives to the viral delivery system. For example, the nanoparticles, which enable scalable manufacturing and flexible cargo. Very important for targeting disease indications that need delivery of longer payload carriers, but of course these do come with some of their own challenges, primarily the efficiency, where the targeting of these nanoparticles become really challenging. Similarly, exosomes, which provide natural targeting and biocompatibility, making them more amenable from immune system point of view, though large-scale production remains difficult. And these systems, while being used for some therapies, they seem to hold the key to the future of repeat dosing and broader accessibility. Somehow, if we can take the goods of nanoparticles and the exosomes or the viral vectors and come up with a new delivery system which will allow specific targeting, low toxicity, the amenable cargo size, carrying ability, all that can kind of like, if somehow we can bring in all those goods, that will create a really great delivery system for these molecules for the treatment of genetic diseases. Okay, so I hope I was able to invoke some interest through these innovative approaches, and moving on to how the future of therapeutics for genetic disease treatments look like. And we can start with AI, which is the latest revolutionary technology changing landscapes of fields beyond biotech. And you are probably hearing AI in everything nowadays. So we, in the same way, we are already seeing how AI is accelerating the drug discovery across the value chain while supporting more efficient target identification, molecular design, and delivery optimization. And all of these are or were used to be the bottlenecks of drug development at various stages of the overall conception of the idea to taking it to the commercialization. So that's where use of AI has helped in expediting some of these activities, such that we can shrink the overall the drug development timeline. Which when I was a grad student, I used to hear it takes almost 15 to 20 years or 15 years from the conception of the idea for the product to be commercialized. Now, through these path-breaking innovative ideas, as well as the technology, particularly in the computers or the AI, that has allowed to shrink this timeline to almost like less than 10 years now in some cases. So in general, while use of the AI for vaccine development is a great example for how this technology can accelerate drug development, similar uses for other therapeutic areas, including genetic diseases, have started to show the impact on the overall development cycle of a molecule from bench to clinic and to the commercialization, something that I was just talking about. And I think like some of us, or a lot of us, are still wondering whether AI will, is it, as the future is moving forward, will AI take over the drug development completely? I think at this point, it's fair to say that AI is a great complement. It potentially, it cannot, or hopefully it won't replace, but provide the biological insight that will allow the faster transition of the programs from one stage to another stage. Okay. The next is the personalized medicine, something that we touched upon in the cell therapies. So the personalized medicine is the next upcoming approach that allows tailored therapies to individual genetic profiles. If there is more widespread and ease of access to early genomic profiling, that will enable earlier diagnosis and targeted intervention. CAR-T therapies like Kymra, which we earlier discussed, exemplifies this approach through regulatory. And as I mentioned earlier, the manufacturing complexity remains high in these kind of modalities. However, more and more effort is being put on the personalized medicines, because the benefits of personalized medicine, specifically for some of the genetic disorders as well as other diseases like cancers, have shown like really high benefits from this kind of a approach. As we move forward, I wanted to spend a few minutes on the regulatory and ethical considerations as well. This becomes important for the treatment of genetic diseases through this groundbreaking therapeutic options. So as we continue to kind of like think about more and more about these genetic diseases and what we are coming up with, the regulatory and ethical considerations become that much important. These therapies, as these therapies will become more and more powerful through additional research, through scientific communities, the importance of the regulatory and ethical consideration becomes that much important to control and put the uh, guardrails for these kind of therapeutics. In general, long-term safety monitoring, equitable access, informed consent, and pricing strategies also must evolve alongside the science. So, as we are thinking about taking this path-breaking science to all different kind of countries, making the standard of care hopefully at some point equitable, if not, well, equitable may be a strong word, but like somewhat similar among different countries. That is what eventually, we should aim for, and that's where building these guardrails through the ethical approaches becomes that much important. And as I showed you earlier, also, the pricing of these gene therapies for right now is pretty high, and how can that be managed in kind of underdeveloped or developing countries is a big hurdle for these kind of therapies to become the mainstream therapy in future. And that's where a lot of regulatory bodies and ethics committees are discussing the best possible strategy for the use of these therapeutics in future. And as a scientific community also, this kind of, when we are building an idea, keeping in mind some part of this the ethical aspects of that science that we are doing becomes is also important, right? Like that kind of grounds us um, in a way where we know that what we are doing should be should have should not have only the a limited scope or limited outreach, but how can we make sure that that science reaches globally? So responsible innovation does require alignment between developers, regulators, and the in general society. And when I say society, I mean to say the governments and like in general the people, how they adapt to that kind of innovation. What will happen otherwise is we will start to see the use of these therapies beyond just the treatment of genetic diseases, where all kind of other options are available for people to kind of like use this kind of science for making superhumans or and whatnots, which again will bring in the ethical considerations of these kind of therapies. Okay, money slide. So how's this therapy growing? And I kind of like, I'm speaking here from a industry point of view, because we need to understand how does this also help not only the patients, but also is there some meat to this overall therapy that will allow continuous investment into the therapy. So as these graphs are showing, the advancement of this technology has allowed the genetic medicine market to grow rapidly in last 10 years. And there are projections. This report that I found kind of has projected, and please, these numbers that is shown in this report, if you go onto the internet, and you can find all kind of different reports on this topic. So I think my advice is the absoluteness of these numbers may vary depending on who is doing who's putting together these reports. But what it tells us through these graphs, which I broke down into the past decade from 25, the year that just ended, and the new decade that is coming up, how does the growth of the genetic medicine market look like? And as you can see, the field is continuously going to grow, and based on this market report, it can become a $450 billion market worldwide by 2035. And that is huge. And this a lot of this is attributed to the amount of work that is going into this field, the amount of interest that this field is gathering, uh, all that because the treatments for the patients are available, and more and more people are seeing the value of this therapeutic area for the treatment of these genetic diseases. So in general, this growth is driven by a lot of work to improve the platform technologies, increasing investment, and expanding clinical success. Of course, the future sustained growth will depend on solving the challenges or the gaps that we earlier discussed, like manufacturing, access, and the cost challenges, which is where the new generation of researchers, clinicians, and entrepreneurs, of course, can help in solving the existing problems and allowing the access of these medicines for global patient population. Okay. So before we kind of like come towards the final slides of my talk, I wanted to through this slide capture the summary of the message I wanted to bring across to everyone. So while there is no single winning modality in genetic disease space, as is obvious from multiple different modalities that we went through, and we saw the challenges or the gaps that there are, the success, however, will come from a combinatorial approach. Innovating new delivery systems and manufacturing technologies are as important as the actual scientific breakthrough that allowed the inception of the treatment of the disease. This is very important, I guess, because as much as I have learned while working in the field, is not all the ideas, the scientific ideas that come into fruition in the early research end up becoming a commercial or a clinical success for that matter. And a part of that prevents that from happening uh, is the actual delivery system, particularly, which is true for these genetic diseases. And as you can see that, taking this cargo of DNA or RNA and making sure that we deliver it at the right location, that is a very complex step in itself. And a lot of drugs or these scientific breakthroughs that are made in early research fail at that very level because it doesn't get delivered to the right organ. Liver is something that we have been able to reach out, which is probably because it is the easy source. Everything ends up in liver. But for a lot of these genetic disorders, which are central nervous system based or the muscular ones, we are still working, or the field is still working hard for identifying the right delivery system that can allow the allow that the cargo to reach to these different organs of the body. Working both on the viral and the non-viral vectors I'm talking about. And that's where maybe AI is something where we can kind of like use that AI that can help us with making it more efficient in terms of like delivery, and also at the same time making more personalized medicines, helping with design of the CAR T cell therapy or any kind of different stem cell therapy that will be more personalized for the particular patient and for the particular disease. And in the same way, when you think about the manufacturing, that is the other big hurdle, because most of these early scientific breakthroughs, they do face a huge hurdle of manufacturability, because what good is an idea if you cannot actually create it or manufacture it at a large scale that will allow use of that molecule by hundreds or thousands of the patients. So that is the other aspect or other hurdle that people are working towards, how to kind of like ensure that all these scientific breakthroughs, or rather most of these scientific breakthroughs, do see the light of commercialization with a great manufacturing plan, which allows production at a larger scale, which means the cost goes low, cost drops, and the outreach to the global patient population becomes easier. So, having said that, what I want to kind of like take home is for all of us is that these are all very intricate topics, and somebody sitting in a research lab doing a scientific innovation, and somebody sitting at a different level, 5 steps ahead in terms of like CMC or the manufacturing innovation. Somehow these people need to come together and collaborate to kind of make sure that that idea that has generated at the bench scale and has value from a clinical point of view can see the light of the actual commercialization. So collaboration across disciplines and organizations will define the future of genetic medicine in general. That is kind of the other message I wanted to send across. So, this is this this brings the snapshot here. I took this picture yesterday. I was scrolling on LinkedIn and I found somebody posted this America's most expensive drugs. And most of these drugs are for genetic disease treatment. And you can see all these 10 drugs are, multi-million-dollar drugs. And the cost associated with the cost associated with these drugs limits their use across the globe. However, I'm sure we have, if once the personalized medicine and the patient identification becomes more and more easier in other developing and underdeveloped countries, we'll see that the disease that they are targeting, there will be patients for these drugs over there as well, but the reach of these drugs for now is limited because of the high price tag. And that's where it kind of is important for the scientific community to come together and identify new ways to either treat these genetic diseases with a more cost efficient medicine, or come up with ideas from a manufacturing or whatever the reason that there is for these to be so high, to reduce the cost burden of the patients, especially for developing and underdeveloped countries. Okay, that kind of was my last slide from this section, and I do have a closing slide before I will open the floor for questions. And in general, I just hope that I was able to pass the message about the future of genetic disease therapeutics, which is incredibly promising and growing at the moment. But as I hope I was able to kind of pass on that message, it does require sustained commitment, collaboration, and responsible innovation. And with that, thank you for your attention. And I really look forward for questions or additional discussion on the topic.
Thank you, sir, for such a wonderful and insightful session on innovation in therapeutics modality for genetic disease. Now, I request participant to ask question in the chat box. So there are two questions.
Sure.
First is from Dr. Saroj. I have been hearing about CRISPR-Cas9 technology for a while now. Can you please update me on when we can expect it to be available as a therapy in the medical industry?
Yeah, I mean, as I highlighted in my slides as well, there is one CRISPR-based therapy already available for the treatment of sickle cell disease. And there are a lot of clinical trials ongoing, which are in the late stage in the clinic in phase 2 and phase 3. So hopefully by establishing through that one commercial therapy that there is the path forward for that therapy, like hopefully the rest of the clinical trials, I don't think all of them will be successful because that's the odds of how it works. But I guess some of those other therapies which are in phase 2 and phase 3, we will hear soon about the commercialization of some of those therapies in future. And by future, I'm not talking about 10 years from now. I'm talking about probably this year and next year.
So the second question is Cascavy CRISPR-Cas9 approved by FDA for sickle cell disease. So this is a question. I think this is a statement or
I'm sorry.
something.
Can you? What is the question again?
Cascavy CRISPR-Cas9 approved by
Yeah.
FDA for sickle cell disease.
Yeah, that's right. Well, I mean, that seems like a statement, not a question.
Another question, what do you
There's
think?
one more
Or
question. Payal is asking.
what do you think or your perspective on biosimilars? Will this category be able to low down the biologic drug for genetic disorder treatments?
Yeah, that's a great question. I think yes. I think my big answer is short answer is yes. The big part of this is I think like we should be keeping an eye on how the patents are coming off and what kind of biosimilars can be made. Particularly, I think like in the RNA therapeutic area, I think biosimilars can play an important role because in general the chemistry is well defined, and these RNA molecules, the manufacturing and all that synthetic manufacturing, so that is much easier than more complex manufacturing for say AAV based or for that matter the cell therapies as well. So biosimilars will play a critical role, I guess, for the treatment of genetic diseases as we move forward in future. And some of the existing the patents will expire, and those medicines can be available for widespread scientific community to kind of like create biosimilars.
So the next question is, and what about the protein therapeutics?
Yeah, well, the thing is, earlier in one of my slides, I highlighted the existing therapeutic field for the genetic disorders, enzyme replacement plays an important role. In fact, the company that I'm currently working with, that is one of the focus areas of the company, where the genetic disorders are treated by enzyme therapeutics. And enzymes, as you are aware, are the proteins essentially. So we already have enzyme therapeutics, or the proteins are being used for the treatment of genetic diseases. The limitation there is that you have to kind of like have the sustained you have to continuously dose the patients depending on how effective the protein or the enzyme is. Meaning, sometimes it's like a weekly dose, bi-weekly dose, monthly dose, and that kind of goes on for the lifetime of the patient. So that kind of puts a lot of burden for the patient, for the overall, the community in general, for the families and all. And also, not to mention, these genetic disorders, these therapies become very costly eventually for the lifetime of the patient. And that's where like more advanced technologies like gene therapy or cell therapy can become options, because they allow one-time treatment. So for example, a AAV gene therapy, you can just like treat the patient one time, and they're good for at least for a decade or so, depending on the durability of the drug.
Ma'am, if you allow, may I ask my question on my own?
Yes,
Yes.
please. Carry on.
Okay. Sure. So to correct my question, like, and what about the protein therapeutic? My question was regarding, like, in previous answer you mentioned that biosimilar development is more possible
Okay.
in case of the mRNA and these RNA kind of therapeutics. But so my question is, like, is it not possible for the protein-based therapeutics? First
Yeah,
part.
yeah, absolutely, absolutely, absolutely. Sorry, I didn't get that question, and I was not able to answer in completion. But yeah,
Okay.
you're right. My mind was on RNA therapeutics because I think the you know, unless like we have figured out the pathways and these protein therapeutics, how the ease of biosimilar generation to me is with the RNA therapeutics. But yeah, no, protein therapeutics can be equally effective from a biosimilar point of view.
And do we really have to wait for the expiration of any patent for developing a biosimilar, or we can explore some other ways also?
Well, I
Like,
mean
is there
that
anything, any case or any incidents you would like to mention over here?
I don't. I'm sorry if you have something to share. I don't recall any incidents where you can have a biosimilar uh, without the patent going off market. But
Okay.
unless like there has been any such case study, I would be more than happy to hear about it.
Okay, okay. No, I also haven't encountered any kind of like
Yeah.
study related to this thing. I was like more curious to understand if something like I can learn from this talk. So
Yeah,
this
no,
is what.
I have not heard about. Yeah, the ideal process or the pathway is, because otherwise the company who holds the patent will always prevent you from going into the market, even though if you maybe your biosimilar is even better. But one thing I can
Okay,
tell on that. Oh, sorry, go ahead.
okay, okay. And like, see if company is working in this domain of development of biologics and the biosimilars, and they keep an eye on the expiry date of the biologics because they already start their biosimilar development process, like
Yeah,
maybe 2 years, 3 years, or 4 years pri- prior to the expiry date
yeah,
of any of the biologic therapeutics. So,
yeah, yeah.
if in that journey they somehow develop a therapeutic which is more efficient than the existing one, so do we consider that therapeutic as a biosimilar?
Yeah, absolutely. I mean, I think like there are regulatory guidelines that define a biosimilar, that one categorize a biosimilar and distinguish it from the existing therapy. So if it is more efficacious, that's where the regulatory guidelines will be more helpful. But they are very clear on like what is a biosimilar, what defines a biosimilar, and what defines a more efficient biosimilar, or at that point does it become its own class of drug beyond the existing therapy. So that can become the class 2 or like the next gen drug. So
So
your
that
your
could
point
be
is valid
considered
one.
as a biologic, new biologic, not
Exactly.
biosimilar.
Yeah,
Okay. Thank
yeah,
you so much, sir. Thank you.
sure. Yeah, but I think like that was a great question and good discussion, because to your point, you can go ahead and do the research, do the development work, make sure, as long as you're not commercializing it and doing your development work, I don't think anybody can come after you. Nobody can prevent. There are no laws, or I don't think there is any restriction on you working on something from a biosimilar point of view.
Hmm. As
But
far as I have
commercializing that
gone,
becomes the hurdle.
because the regulatory guideline which is available in India, which is quite pioneer, because earlier the first guideline was provided by the Europe, and the next was adopted by CDSCO, and later by the FDP. So I have gone through the guideline and some of the queries which I found. So I'm just like looking into it, and I'm already going through this topic and following this thing from last 2 3 years. So I'm more curious. So that's why I asked. But thank you so much
Okay.
for all the doubts cleared.
Yeah, yeah, absolutely, absolutely. And I hope I was able to answer your questions clearly.
Yes, yes. Thank you so much. Thank you.
Any other questions?
Yeah, there's one more question on the chat box. So Dr.
Sure.
Saroj is asking what advancements can we expect in small molecule therapies, and how will they impact future treatment options for complex diseases?
Yeah, that's a good question. I mean, I think like small molecules is we continue to evolve the chemistry of small molecules and make them more specific, targeted. So that in that sense, I think we can kind of like where we can see the growth is because right now I think the when we look at the PKPD results in the animal studies or even in the clinic, I think there is a room for improvement. There are major challenges in small molecule chemistry in terms of like the off-target effects. Uh, and in general that kind of like leads to um, higher doses that you have to give to patients because of its non-specificity, and therefore that brings in the burden of safety, as well as you have to give the drug at a higher frequency to the patients. So I think to answer your question, there is from a from a design point of view, improving the specificity of these small molecules and improving their pharmacokinetic properties, which will allow for longer half-life of those molecules in the blood, which therefore reduces the frequency of dosage and the amount of dosage for the patients, and hence reduces the side effects in the patients. Those are the things I think like will continue to improve in small molecules. The good thing is, the manufacturability and everything is kind of we have done a lot of work in that field, so we kind of have a good idea for these small molecules, especially because most of them are synthetic. So I see the focus more towards the design, and that's where the AI will be really really helpful, because we can kind of like go beyond uh, just that it can improve our rational design when we are in the early stages of the program, where we can test like hundreds or thousands of compounds and see which one is the better hit, more targeted hit for the disease indication we are going after. Does that answer the question?
Yes, she's saying thanks.
Okay.
Any other question we have? I think you have extensively covered each modality, advantages, disadvantages, everything. So it was a well structured and detailed talk on these modalities. Just one question, because we are in India, so like what modality is more like common here? Any idea, like for genetic
I think
diseases?
I think protein for genetic diseases, I think in India we are relying on protein biologics or enzyme therapeutics, as well as small molecules, which uh, specifically they don't, like, say for example, small molecules don't go to the root cause. They're basically treating the symptoms, not the cause, the root cause. And that's kind of something that we are very much focused, and that is because of the accessibility, the cost, and everything, the burden that we have over there. Similarly, protein biologics, to an extent, they can be treated, they can be used for the root cause as well. For example, if somebody is missing an enzyme, a good example of that is the treatment of hemophilia. So, hemophilia A and hemophilia B, which is currently being used, the existing therapies are providing the factor VIII protein to these patients. However, again, the patients in these cases, they will still have those bleeding events for hemophilia, because sometimes if they miss a dose or something happens in between, that just makes them prone to bleeding issues. And therefore that's where like something like a gene therapy can become more advantageous, because that's like one-time treatment for these patients. But yeah, to your question, Dr. Shilpa, I think small molecules and the proteins are currently what is being used in India, primarily because of the ease of access, cost effective, and widespread availability across the country as well.
Thank you so much. I think we don't have other questions. Thank you for taking out your time, and you are so kind and considerate getting early in the morning. And thank you so
Yeah,
much.
no, I enjoyed. Yeah, thank you for the invite, Dr. Shilpa. I know we have still some time left, but I thought I will keep it the sessions within one and a half hours, such that we can have more time for discussion. So really thankful for first the invite, then the questions, and you are very patient host, because I did kind of like left it to the very end, some of the things that you requested me. So thank you so much.
Thank you so much.
Okay.
So
All
with
right,
that,
we'll sign off.
Yeah, thank you
Sorry,
so
go ahead.
much. And with that, we end our day 2. So I'm posting the attendance links in the group, in the chat box. Those who missed filling up the form, they can fill the day 1 form. And many of you are asking about WhatsApp group. I have already given the link of WhatsApp in the email. So you can join the group there. Thank you. Thank you, everyone. And tomorrow our session will start at 6 p.m. Thank you, Dr. Vikas.
Thank
And
you.
thank
Thank
you,
you, Dr. Shilpa.
everyone.
Have a nice evening. Bye.
Okay, good night.